You sign into Zoom and then you start the webinar. It's login with Google. Login with Google? Like sign in with Google? Instead of putting in the email password, it's oh no.
Okay, if you guys can see us and hear us over in Zoom, give us just a couple of minutes. We are setting everything up for you. Hold tight. We've got raised hands. Hold on just a second. We are setting everything up for you guys. I promise. Okay, I see comments in Crowdcast that you guys can see me in Zoom. So that is awesome news. Just hold tight for a little bit. We've got to set everything up for you. Yeah, lots of comments in Crowdcast that you guys can see me here. So perfect. Give us just a couple minutes. We promise we'll catch up. If we don't get through everything tonight that we had wanted to, it's totally okay because we did set aside extra time in future nights so that we can just in case there's problems sometimes, and it's totally okay. But I promise we're gonna get everything set up for you guys. Just hold on. So I'm gonna mute myself, but I'm still gonna be here so you guys can see me. So yeah, just hold tight. Again, if you are just joining Zoom, give us a couple of minutes. We are setting everything up for you. I promise. We're working on getting this all. I know Crowdcast sometimes has issues, and we want to make sure to get this all set up for you guys. So just hold tight. All of our hosts, almost all of our hosts are here. We're waiting for Sean. Sean needs to be added. And we'll get there. Just give us a couple of minutes. For our hosts that are here, can you guys review? I know we don't have our slides up yet, but we'll get those back up. Where we started with crashing, can you guys go back and start reviewing that as we work to get slides up and everything?
Yeah, I couldn't see
Amazing.
the
Thank you.
chat. Is that where we were?
I am not sure exactly where we had left off, but I know
I
it was
think
over
you-
in Neurons somewhere.
I think this is where
Okay.
we lost people, was on this slide.
Got
Johnson!
I'm here. I made it.
Amazing.
Okay, can
Are you
everyone see my screen?
sharing?
What was that?
I'm not sure who's sharing their screen.
Can you see it? That's my screen up there.
Okay, can you go full screen on the slides?
Oh, you can see my whole screen. Haha.
Yeah.
Wait, give me 1 second. Let me just share it as a tab.
Okay, perfect. Thank you. The
See
chat
all
is
my-
disabled on Zoom. We will try to figure all of that out. Hosts, you guys can leave Crowdcast. I'm gonna keep Crowdcast up because it looks like you guys are still chatting in Crowdcast. If hosts want to do that as well, you guys certainly can. And then because it says Zoom host or Zoom chat is disabled, we can keep chatting in Crowdcast if we want to do that.
Thank you guys so much for your patience, by the way. You know, Crowdcast, it happens to the best of us. There's nothing we can do with technologies, but we're gonna get you guys all the AP info that you need. Don't you worry. Don't worry for a second.
Like I said earlier too, we added extra time and a lot of time for technical issues, or if we moved slower through certain content. So promise you guys we'll get everything taken care of for you.
Okay, who's sharing the slides? Because I can start going back over the
So,
neurons.
Sean's just
or-
sharing his whole world with us right now.
yeah, right.
Oh no.
Okay. Are we good?
Yeah, looks great.
Oh, I can't see anything.
Rhea, you can't see the slides?
No.
Victoria,
Chat.
because I can see slides. Victoria, can you see slides? Or Jessica, can you see slides? Okay, you guys can see slides. Chat over in Crowdcast. Can you guys see slides? Enable the chat would be easier. Yeah, I have no idea. I don't have the technical control over the chat, the Zoom chat. But it does look like our students can see the slides. So I think we are ready to go. We will do our best to kind of work through everything as we go. But yeah, go ahead and take it over.
Rhea, are you still able to see not able to see the slides? Okay, I'm not getting anything from Rhea. Jenna, would you mind maybe taking over this section? I know the brain is your thing, so
Heck yeah. Yeah, I love it. Oh my gosh. Okay, I wasn't prepared to host, but I'm so ready to do it. All right. This is the neuron. All right, so when we're looking at the structure of the neuron, there are a few important things to note. The neuron, if you hold your hand up like this, you can make your own neuron. So we have dendrites up here, soma, cell body, and travels all the way down to the axon, down the axon to the axon terminals. No joke, I had a student in class one time during a test, taking their test with their arm up like this, going like dendrite, soma, and they were doing the whole thing. So that is exactly how you can remember it. So dendrites. Dendrite is like a tree. So if you think about the word dendrite or something that's dendritic, it means, vocabularily speaking, it means tree-like. So that's what we're looking at with dendrites. Now I know that the axon terminals and the axon bits at the very end also look tree-like. So I know that that can be confusing, but the important part about the dendrites, differentiating them from the axon terminals at the end, is that the branch-like things come off of the cell body. That's what we're looking for when we look at a neuron. So we have our soma / cell body, the tree-like dendrites coming right off of that, and then traveling all the way down the axon. So that long spiny thing, that's called the axon. And then that pops into the axon terminals at the very end. They're kind of like a burst. So what happens is with the axon, we have this protective insulating cell that wraps around it to speed it up. This is called the myelin sheath. Now I will ask the Crowdcast chat that's back here to tell me, what is the myelin sheath made out of? There are, like I said, that there are cells that wrap around the axon, but what type of cell? Okay, people are saying like fatty substance, which is true. Can anybody name the type of cell that wraps around to create the myelin sheath? So fatty cells, perfect. That looks great. Also, somebody mentioned Schwann cells. That is amazing. Yes, that is correct. Glial cells, that's also a word I would be looking for. And then another one that I would be looking for is oligodendrocytes. So Schwann cells make up the myelin sheath in the peripheral nervous system, oligodendrocytes in the central nervous system. And then again, we've got our axon. The components between the myelin sheath are called nodes of Ranvier. And then we have our axon terminals at the very end. I like how somebody wrote giglial, like we're giggling. Super cute, love it. And then we can create a synapse. So a synapse is the space between the axon terminal of the presynaptic neuron and the dendrite of the postsynaptic neuron. So it's this little tiny gap that's made between the connection points here. They don't actually physically touch, they create a very, very small gap across which neurotransmitters are passed. So I'm good with this section. Is there perfect. Do we have who was originally doing this? Was it Ria or was it Jessica? I just want to make sure that I've got everybody where they need to be.
Ria was going to be covering our main Unit 2 Neuro Girly. She still seems to not be here, but I am happy to hop on as well.
Yeah, we can all just tag team this. Let's just do that. So Jessica, Victoria, feel free to unmute yourselves and we'll talk about this as we go. Somebody's mom says it's made out of plastic. What is made out of plastic? I have no idea. Oh, can you define the nodes of Ranvier? Yeah, the nodes of Ranvier are just the gap between the little glial cells that make up the myelin sheath. It's where the action potential occurs. So it's an excellent lead in to getting to the second part of this slide, which is the action potential. But first, the resting potential. Jessica, do you want to talk about the resting potential?
Sure. So the resting potential is the phase that is going to be occurring in the cell before the action potential. You need the resting potential for the action potential to occur. The resting potential voltage, the inside of the cell relative to the outside is going to rest at between -70 and -90 millivolts. I'm going to spare you a ton of biology right here, but the two main ions that are really important for the resting potential and action potential happening are sodium and potassium. Sodium is going to be concentrated a lot more on the outside of the cell than on the inside, and potassium is going to be the opposite. It's going to be concentrated about 30 times more inside the cell than the outside of the cell. So just by like basic 9th grade forces of diffusion, the cells that are concentrated, or the ions concentrated inside the cell, are going to want to go out, and the sodium on the outside is going to want to come in. And this sets up a concentration gradient that is important for the action potential when it's triggered.
Amazing. Thank you so much. So speaking of the action potential is the neuron firing. So that's really, really important to send any type of signal. So realistically, when we think about all of behavior, thought, conscious experience, feeling, emotion, all of that is coordinated by the action potential of neurons. So this is happening super, super fast. They are very, very rapid. Importantly, axons that are myelinated, they actually have action potentials that are faster. And that is because the action potential only occurs at the nodes of Ranvier instead of occurring along every single segment down the axon. So with our action potential, there is a threshold, and this threshold is a resting threshold that we have with the resting potential, and then there is the threshold of excitation, where as signals come in through the dendrites to the soma or the cell body, they kind of accumulate, they kind of add up, and if the cell body gets the addition of or a summative amount of certain, I'm like thrown now because I was not expecting to teach this. If there's a summative amount that reaches that threshold of excitation, it raises the millivoltage of the neuron to that threshold. What happens is the action potential will fire. If the threshold is not reached, so the summative millivoltage changes come in, and they do not reach that threshold, then that excitation just kind of like what's the word I'm looking for? Like diffuses out, and we don't have an action potential that happens. If the threshold is reached, then we do have an action potential. It's all or none. It either happens or it doesn't happen. With action potentials, they're not sometimes stronger and sometimes weaker depending upon the impulses. They're always the same strength. Sometimes you'll have more, like sequentially more action potentials versus sequentially fewer action potentials. And that can translate to the intensity of a stimulus. For example, if you just rest your hand on a table, that would be fewer action potentials within a period of time compared to if you slam your hand on a table, that's going to be more action potentials in a like amount of time. Somebody's asking, what is MV? MV stands for millivoltage. It's just the way in which we measure the amount of the volts that are coming in. Like we said, these are all or none. And what happens is that ions, positive ions, are going to flood that section of the neuron. And when that happens, that will depolarize our neuron. So it becomes less negative and going towards positive. That fires the action potential. And then in order to end the action potential, ions are going to flood out of the cell. So the positive ions leave the cell, and that is going to repolarize our cell. So you see it like the millivoltage starts here, reaches the threshold, goes all the way up. That's hyperpolarization. And then it depolarizes. We actually end up in a hyperpolarization. And with hyperpolarization, basically more of those positive ions leave the cell than initially had flooded in. And this creates a neuron that is more negative than it initially was in the first place. This hyperpolarization creates what's called a refractory period. And a refractory period is a moment in time when that neuron simply cannot fire again because it's even more negative than it was to start. So yeah, that's it about the action potential. Moving on. I think I saw that Ria is back too. So if Ria wants to pop in as well, you certainly can.
Yes, yeah. Sorry, I don't know
Welcome
what was going on. Hi. I like couldn't rejoin for some reason. That was stressful, but I'm back. I'm back. So for neurotransmitters, these are the things that are released by vesicles at the end of the axon by the axon terminal. And so once the action potential reaches the very end, it's going to trigger the release of them. And one really important ion for this process is calcium. So calcium is very involved in letting the vesicles be released into that synapse. And so psychoactive drugs can affect the release of neurotransmitters. Agonists can affect it by increasing the action of it. So an example of this would be like SSRIs. These are selective serotonin reuptake inhibitors. They increase the amount of time that serotonin spends in between the axon and dendrite in that synapse. So then it's like more likely to be taken up by the dendrite in the next neuron and continue its reaction. But just always remember the agonists increase. And then on the other hand, we have antagonists, and these will decrease the action of a neurotransmitter. So an example of this would be like Botox. So when you get Botox, it's blocking acetylcholine receptors. And if y'all remember, acetylcholine is necessary for muscles and muscle movement. So if you're blocking acetylcholine receptors, you're not able to move those muscles. So that's why some people will get Botox in their face. And so if you get like Botox in your forehead and you're blocking the muscles from acting there, you're not able to use acetylcholine, you're not going to be able to move it. And so that's going to help you like avoid wrinkles. But the main underlying process for that has to do with acetylcholine and that neurotransmitter. So then moving on to the next slide, we're going to summarize a couple different neurotransmitters that are pretty important in AP Psych and their functions. So like we talked about in the last slide, acetylcholine is vital for muscle contractions and muscle movement. And this is a specific neurotransmitter that's targeted by black widow venom. And that's why like people can't like control their bodies anymore. It's just going crazy. And then so it's involved in muscles, learning, and also attention. And then in Alzheimer's, that's when you have a decrease in the amount of acetylcholine. And so the acetylcholine producing neurons die and you're unable to like move as well as you could. And this can lead to things like tremors. And then we have glutamate. And glutamate is the main excitatory neurotransmitter of your nervous system. And one way I like to remember this one is like it has the word 8 in it. And every time I eat, I get excited. So it's like glutamate, excitatory. And it's also pretty important in learning and also your long-term memory. And then we have dopamine. So this is involved with your reward system, with movement, with motivation. And I remember this one, you sell dope at the park. Dopamine has to do with Parkinson's. Deficits in dopamine will lead to Parkinson's. And then ADHD and schizophrenia can do with other imbalances. Schizophrenia specifically, it's when you have too much dopamine and you can't focus. It's like just your reward system is completely out of whack. And then for norepinephrine, this is all about fight or flight, and it has to do with alertness and attention. And anxiety can happen when you have a prolonged, like too much norepinephrine in your nervous system. And then depression can be when you have too little. And so norepinephrine, its effects are like vital for that small period of time when you're like going under stressful circumstances. But when its release is chronic, when it's over a long period of time, it can really affect you. And that's what leads to things like anxiety, when it's like the elevated heart rate or that stressful mindset for too long, and you just can't sustain that. And then we have GABA, and I remember this one, like when you listen to somebody gab, you want to go to sleep because GABA is a main inhibitory neurotransmitter and it's involved in sleep. So when it's released, it's inhibiting your muscle movement and it helps with you like staying asleep. And then epilepsy and insomnia are both some examples of what can happen when there's dysregulation in this system. And then we have serotonin. And serotonin is all about mood regulation and depression. And then if you want to remember 4 of the specific functions of it, if somebody can type this in the chat, because I know y'all like love the mnemonics, but Ninja Turtles live in the sewer or SUA. It's SEWA. So serotonin involves sleep, eating, wakefulness, and aggression. So that's an easy way to like remember its functions. And then on the next slide, we can talk about drugs and how they affect your brain. So a drug is anything that can lead to changes in your perception or mood. And there's depressants and there's also stimulants. So for drugs, it's like very different ways that it'll interact with your nervous system and your brain, but they can have similar reactions. So for example, tolerance, that's when your receptors change. You can have less of a response to the same drug. The same dose that you once took will have a lessened effect. So an example of this would be like somebody who like drinks alcohol. Like when you start drinking, 1 cup of wine might make you like have a certain reaction. But then the longer that you continue drinking, like over the span of like weeks or months or years, that 1 cup of wine will not have the same effect that it used to. You might need 5 cups of wine to have that same reaction that you once got from just one. And so exactly, like a higher dose is needed for that same effect. And this is what is really dangerous for people with opioid addictions because, or any addiction really to drugs in general, because you'll increase your tolerance. And as that increases, if someone chooses to go through like the recovery process and they stop using it, if they happen to like start using again and they try to use that same initial amount that they did like right before they went into recovery, their body can't handle that much anymore, and that can lead to death. Like it's very interesting how your body can up and down regulate receptors and neurotransmitters depending on what you're using and what you're putting into your body. And then kind of continuing this process from tolerance, you go into dependence. So that's when your body relies on the substance for normative functions. So you might have like a baseline of dopamine in your system that your body is like used to. But once you become dependent on a drug, that baseline, because you're introducing this external substance, it's now increased. So if you were to stop taking the drug, you can't get to that upper level anymore without that outside help. So you have a new like dependence on this drug just to reach your new baseline. And some people can get issues such as body aches, fevers, chills when they are not able to reach that threshold anymore. And then from dependence, we go into addiction. And addiction is a chronic psychological disorder. And you're having compulsive drug seeking behavior and drug use. you spend your time thinking of how you're going to acquire a drug, using the drug, recovering from using the drug. It's that whole process, but it's when that dependence you can't function anymore normally without that drug. You need it just to be baseline. And then moving on from that part of the process, we go into withdrawal. So this is when somebody stops using that drug, or they're not using as much as they used to, and their body is trying to kind of battle with the fact that you're not able to reach that baseline anymore, that new baseline that was set when you started using the drug. And some symptoms with this are shaking, vomiting, nausea, anxiety, cravings. It's your body telling you something's wrong because it's not at baseline. But that's why people who are going through recovery need that extra help to help them get through this process and hopefully lead to complete recovery. And then we have a practice multiple choice question for y'all on the next slide. So y'all can answer in Crowdcast if you're still in there, or just think to yourself, but an individual who drinks alcohol daily will find it necessary to drink increasing amounts to achieve the same state of well-being attained in the past. This is an example of, so I'll just wait a second. The poll is on Crowdcast, so y'all can answer on there. But the answer is alcohol tolerance. So it's kind of what we talked about before. Is anybody on Crowdcast? Can you see how people responded? Like, did we do pretty well? I don't have it.
The answers are still rolling in, but the answers that we have gotten from the poll are really, really great. So it looks like you guys are getting this, which is awesome.
Awesome, that is great. So then we can keep moving then. So this is when we're going into kind of the bigger anatomy of the brain. We have your left hemisphere, which controls the right side of your body. And the left hemisphere is mostly involved with language, reading, writing, speaking, interpreting information, math, and logic. And then we have our right hemisphere, which will control the left side of our body. So everything is kind of flipped. And the right hemisphere will control your visual processes, spatial awareness, facial recognition, and it also helps with expressing and perceiving emotion. And so within your brain, you have your corpus callosum, which is the tractive axons that's between the two hemispheres, and it's what connects the two sides and lets them kind of talk to each other. And then you have your, I can never say this right, gyri, y'all know what I mean, but it's all gyri, sorry y'all. But it's the cortical fold. So these are the peaks in your brain, and it helps you to increase your surface area. But these are kind of the other part of them are the, Jenna, I'm gonna need your help again. The sulci.
Yes, gyri and sulci, that is the plural, or we would say gyrus and sulcus for the singular.
There we go. I'm sorry, y'all. I'm used to just reading it out of a textbook. I never have to say things out loud, but you know what I mean. So for these, they're the cortical folds. They're the divots, the valleys of these folds. So they have the same function of increasing surface area. And the way that I remember these is that when you're like sulking, when you're sad, your shoulders kind of droop and your head's kind of down. And so the sulci are the folds. And then we have the fissures. So these are the really, really deep sulcuses or sulci between the different regions in your brain. And we kind of use them as boundaries for the different lobes. So if we go to the next slide, we can break that down a little bit more. So we have our frontal lobe, which includes Broca's area, which is involved in speaking, and also your planning and judgment is kind of coordinated by your frontal lobe and your prefrontal cortex. And then we have movement, which is coordinated by your motor cortex. But all of these are in your frontal lobe. And then in your temporal lobe, we have auditory processing, semantic processing, which is all about meaning, and that's in Wernicke's area. And then we have your long-term memory, which is also processed here. Then in your parietal lobe, we have sensory processing through your somatosensory cortex, as well as spatial awareness. And then in your occipital lobe, it's visual processing. So some ways I like to remember these. One thing with Broca's area, Broca, it has to do with speaking and speech, and boca in Spanish means mouth, so Broca, boca. And then with your like temporal lobe, your temples are right by your ears, and your temporal lobe has to do with your auditory processing. And then occipital lobes, occipital is like eyes, it's vision. So going to the next slide, we're going to dive a little bit more into the neuroanatomy. So starting with the amygdala, it's involved in the limbic system, and it has to do with fear and aggression. And so your amygdala is connected directly to your hippocampus, and they both kind of work hand in hand because any fearful or like emotional encounter that you have will be encoded into your memory through your hippocampus. And one way I like to remember amygdala, I think Amy is emo, because it's emotional. And then hippocampus is all about memory, specifically episodic memory, and consolidating things together. And if I saw a hippo on campus, I would remember it. So that's how I remember those two. And then we have our thalamus, and this is kind of like the post office of your brain. It's your sensory switchboard. So all the sensory information you're receiving from the world is going to go through here before it's redirected through your brain. Everything except smell. Smell has its own separate bulb, but it's different.
Ria, can I hop in for just a second? I just have a quick question I want to raise to the people who are still in the chat on Crowdcast, because I've seen a couple of comments and questions about asking if we can slow down a little bit. So we have two options. We can go through the content at a slower speed, so that you can absorb a little bit more of it. But what that does is, because of the issues that we've had with Crowdcast, it has certainly pushed us back in time. So if we do that, what happens is that we might not get to other things that we wanted to talk about. So option A would be we slow down. We talk about some of this content at the speed we initially would have wanted to, but we might not be able to talk about other things. Option B is we keep this review of this section kind of quicker. And then we can still get to the things that we wanted to, at least more of them that we wanted to. So let us know over in the Crowdcast chat what you would prefer. Do not slow down in all caps. Don't slow down. Please don't slow down. All right, I am hearing don't slow down, so let's keep it up. Thank you. Ria,
All
did you
right,
want to pop back? Oh, perfect. Sorry.
seem to have lost Ria, but that's okay. I can pretend to be Ria for a little bit. We were just talking about the thalamus and how it was essentially like the post office. I think that was a really great analogy. All of the senses synapse in the thalamus before going and entering conscious awareness in your brain, except for smell. Except for smell. The hippocampus is really tied to episodic memory. We're going to get to that a little bit more with unit 5 in our cognitive psychology. You want to know that the hippocampus is located in the medial temporal lobes, and damage here will basically decrease your ability to encode and store episodic memories in long-term memory. The hypothalamus also does internal regulation and is tied to that autonomic nervous system that we were talking about earlier. Some of the subcortical structures that are important to know for this neuroanatomy are the medulla, which do internal regulation as well as vomiting. If it senses that there is something potentially toxic that you have ingested, it will take care of that. The cerebellum is important for voluntary movement and procedural memory. It's really important for allowing you to maintain your balance, not to be confused with the motor cortex that initiates movement. The cerebellum actually can't initiate movement, but it's important in coordinating movement. The pons are another subcortical structure that are important to know. Like the cerebellum a little bit, they're important for coordination as well as sleep and sleep regulation. And finally, the reticular formation helps us feel aroused and helps us direct stimuli. All right, I think that we are good to move on to this practice question. Okay, so if you're still in Crowdcast, feel free to answer in the poll or in the chat. The brain scans of people with amnesia are most likely to show damage to the what? I talked about it a little bit in the last slide. All right, I am seeing some answers in the chat in Crowdcast. Great, we can reveal the answer. You guys got it. It's the hippocampus. You're going to have trouble encoding new memories and possibly recalling old memories too. Retrograde and interrogate amnesia. Okay, I think we can move on to our next topic, which is neuroimaging. And if anybody would like to come down and chat it up with me as well, it looks like we've lost Ria, but that's okay. I can be Ria.
I can certainly do that as well. Hey, Jessica.
Hello. So there are 4 types of neuroimaging techniques that we want you to know. They each have their own benefits and drawbacks and have their own, they're each good at different things. The EEG has really great temporal resolution, but not so great spatial resolution. You can't really see exactly what's going on. The EEG is going to be the top left picture where you see all of the little nodes attached to your head with the gel on it, picking up event-related potentials. It's going to watch for and amplify electrical waves, potentially while you're doing different behaviors, and looking at the action potentials that your neurons are experiencing.
Perfect. That's what I wanted to point out too, so that we could relate it to the things that we've already learned, is that an EEG measures action potential. So what it's looking for is that depolarization that we talked about earlier. When a cell or when a neuron depolarizes, that's what an EEG will pick up. So it's looking at the areas of the brain that are firing. And like Jessica said, with the temporal versus the spatial resolution, temporal means related to time. So with an EEG, we have really excellent time-based information. So we can say that exactly at this time, this is the area that was the general area that was firing. But when we talk about spatial resolution, we don't have great spatial resolution on an EEG because we only have a certain number of nodes at different regions. So we can say this is the general area where we saw an increase in activity, but we can't really pinpoint it down to a very specific location. But now we have these other types of radiographic techniques that will be really effective for getting spatial resolution, but maybe might have worse temporal.
Yeah, and actually one of those techniques is the MRI, or specifically the fMRI. Sort of the opposite of the EEG, the fMRI has really great spatial resolution, but not so great temporal resolution, or being able to tell what's happening at the exact time in the exact place of the brain. That's because there's a little bit of a lag with the way the fMRI works. Functional MRI, unlike a normal MRI, shows you both the structure of the brain as well as to some extent the function and what's going on with the different brain parts. The normal MRI will just give you like a sort of a very beautiful picture of the brain using magnetic fields and radio waves. Functional fMRI is a layer on top of that. It uses hemoglobin to track the oxygenated blood in the brain to see where blood is flowing to, what exact areas of the brain are active, and that's why it gives us such great spatial resolution and is able to help us locate which areas of the brain are active during which behaviors. And finally, our last neuroimaging technique is going to be the positron emission tomography, or the PET scan. But the one drawback,
love those.
Yeah, the one drawback with this is that it's a little bit costly because it requires that you make the specific radioactive substance, but it operates using or detecting radioactive glucose to give you that scan of the brain and to see, it really tracks the metabolic functions of different areas of your brain.
Perfect. I'm like trying to like multitask right now, trying to manage all of our like issues that we've had. As a note for everybody that's in the Zoom right now, Crowdcast has had widespread issues tonight. Every single stream is having issues, so it wasn't just our stream. We're really, really sorry about it, and we're doing our best, and hopefully this makes up for it, but we're really sorry. We're gonna go an extra 15 minutes. If our hosts have to leave at 10 Eastern, that's totally okay. But I'll stay on. I know Sean will stay on, and if our hosts can stay on, perfect. And we do have this recording again. I'll figure out how it's getting to you guys. It might just be emailed. So just to kind of soothe all of those concerns.
Great.
Ria is back too. If you want to jump in, Ria, go for it.
The stage is yours.
Hi y'all, sorry about that again. I really don't know what's going on. I keep getting kicked out of everything. But thank y'all for helping out with that. But so for sleep, the main sorry.
I think that we're on a damage first, but sleep.
Damn it.
I love sleep. We'll get there.
So for damage, like I kind of talked about earlier, the main ways that we know what brain regions are associated with what functions are through case studies of these like extreme examples of damage. So for like Phineas Gage, he was an example of personality changes and the function of the frontal lobe in higher order thinking. So he was the guy, y'all should put it in the chat in Crowdcast if you remember, but summarize him for me while I'm talking. But he had like a pole go through his frontal lobe and he had damage and he was unable to kind of function, like his personality was changed through that process, like through that happening, and he couldn't like make very rational decisions anymore. He was much more impulsive. He didn't really have impulse control, and all of that was caused through that frontal lobe damage. So he's one example. And then we have split brain patients, and these were people who had their corpus callosum, which, if y'all remember, we talked about in the neuroanatomy. But the corpus callosum is a part of the brain that connects the left and right hemispheres. And this is what helps you kind of have both brain regions talk to each other. And so for people with this who had this lesion, they, as if y'all remember, like your right hand is controlled by the left half of your brain, right? And your left hand is controlled by the right half of your brain. So when people were given tasks with their corpus callosum split, since our language is in our left half of our brain, when they saw something or touched something with their right hand, since their left and right are connected, they were able to say what they saw. However, when they touched something with their left hand and they were asked to like say what they were touching, like verbalize it vocally, they were unable to do so because since your left hand is connected to the right half of your brain and language is on the left half of your brain, you're unable to say what you were feeling because your two brain hemispheres didn't have any conversation going on between them, if that makes sense. So if y'all need me to re-explain any of this, say it in the Crowdcast chat. But that was basically the gist of split brain patients. And then we have lesioning, and a lesion is any abnormal change or damage to tissue in your brain through disease or through trauma, like in the case of Phineas Gage. But so lesions are whenever there's like a cut or any sort of like damage and we can see what goes wrong, and it's more of a general term. And then for TMS, that's temporary lesioning stimulation. So it's like kind of seeing what would happen if you were to lesion this portion of the brain without actually having to do permanent damage. So yeah, split brain stuff is spooky. But moving on to the next slide, Pep has a question. What are the implications of these studies for neuroplasticity and recovery? So y'all can answer this in the chat. I will answer it pretty quickly, like just talking. So for cases such as Phineas Gage, he had that like hole through his head, right? He wasn't able to recover from that. Like he had permanent damage afterwards. Same thing with split brain patients. Like once you cut that corpus callosum, you're not really able to have neuroplasticity like help you to recover from the effects of this lesion. And so it's really important in that these issues show us that your brain has pretty limited abilities in regenerating and like fixing itself from major damages such as these. And then moving on to the next slide, we can start talking about sleep. So like Jessica said, I feel like we all love sleep, but it's really, really important for us in that it helps us to protect our bodies. It helps to enable plasticity. It helps with growth, memory consolidation. So sleep is vital. And there have been experiments that have shown, like on rats, that when animals are not allowed to sleep, that leads to death. Like you can't function without any sleep. So, for the different sleep stages, we'll start with stage 1. So this is non-REM sleep, and it usually lasts 5 to 10 minutes, and it's light sleep, and it's characterized by theta waves. And then after this stage of sleep, you're going to go to stage 2, and stage 2 is non-REM sleep, and as well, and it's a stable transition stage between 1 and 3. And it's usually like 10 to 20 minutes, and the brain waves associated with it are sleep spindles. And then we have stage 3, and this is non-REM sleep as well. And it's your deepest sleep. So this is a stage of sleep when you're like trying to wake somebody up and it's like impossible. They're probably in stage 3. And during this stage, it's when growth hormones are released, and kids will often have night terrors during this stage. And then the sleep waves, the brain waves associated with this one are delta waves. And these are during this stage, your heart rate will decrease and your breathing rate will decrease. And it's just very like low function. And then during stage 4, this is your REM sleep. So this is when your brain waves are the beta waves, and it's almost as if you're awake because it's the same functionality or the same brain waves you would see in an awake, like active person, but your body is paralyzed. Like you're not able to move or feel anything. And so it's like it's the paradoxical stage. That's what people call it. It's when you're internally active, but you're externally paralyzed, so you're not able to function. But during this stage, you have dreams and memory synthesis. And the main system that's kind of involved in making sure you don't move or feel anything is the reticular activating system. And we'll kind of talk about that a little bit later, but we don't need to know that in too much. But the main idea for sleep is that it's governed by your circadian rhythm, and that has to do with your SCN, which is your suprachiasmatic nucleus, and that's a portion of your brain that is triggered by light. And so when it's dark, when there's no light, that's going to tell your suprachiasmatic nucleus to tell your pineal gland to secrete melatonin. And if y'all remember, melatonin is the hormone that is involved in sleep. So whenever there's no light, melatonin secretion will increase. Whenever there is light, melatonin secretion will decrease. So that's like as like the light will come into your room in the morning, you're gonna slowly have less and less melatonin secretion and you're gonna wake up. And then for the sleep waves, the cycle, the pattern that it kind of goes through in the night, you'll start at stage 1, then go to 2, then to 3, then back to 2, back to 1, and then you'll go to REM sleep. And you're kind of just continuing this pattern throughout the night until you wake up. And the longer you sleep, the further it is into the night, the longer your REM cycle will get, the REM sleep will get compared to the other stages. So then on the next slide, we're going to talk about dreams. So last night I dreamed I was a bottle of ketchup. Like dreams can be pretty nonsensical. They don't always make a lot of sense. And there's a couple different ways that people think about like our dreams and why we have them. One way, I mean, we're back with Freud. I swear, he's literally everywhere. But he said we have manifest content. And manifest content is what you actually remember, like the storyline of your dream, the stuff that doesn't really make sense, you know? So for an example, if I had a dream that I was like being chased by these like wild ducks, like that would be the manifest content of my dream. But then Freud also has something called the latent content, and that's the underlying meaning, like if you were to break it down and read between the lines, what that dream could mean. So an example for like if I was being chased by geese, that could be symbolic of like me being afraid of like a deadline that's creeping up on me. Like I'm being chased by that. That's something that Freud could psychoanalyze and see as a cause for me having that content in my dream. And then Freud also thought that negative emotional content and failure and sexual dreams, these are all things that are like it's just covering an underlying desire or things that are going on in your mind. It's just kind of being like manifest in a different way. And so then why do we dream? So wish fulfillment, manifest and latent, that's Freud once again. So that's his theory on it. But then some other reasons that we dream could be information processing. So all the random things that happen throughout the day, when you dream, that's your time to kind of like break it down and just process everything that happened to you. And then also with cognitive development, because sleep is pretty necessary in forming those your synaptic connections in your brain. That's why when you're studying for a test, it's really important that you get a good night of sleep before. You can't stay up cramming because you don't give your brain time to kind of make those connections between your memories and content. And then there's a physiological function in sleep in that you just need to rest. Your body, like we said before, in stage 3, that's when the growth hormones are released in your body. And so you just need to give your body time to get through all the stages of sleep and make sure that you're physically taking care of yourself. And then there's the activation synthesis theory. And this has to do with the activation of the brain regions during sleep and the random nerve impulses that will be going to these different regions. and your cerebral cortex. So these random impulses don't really have a reason for them, but your dreams are your brain making sense of these random stimulation being sent throughout your brain. And then on the next slide, we can go through some sleep disorders. So, the one pretty big one is insomnia. And insomnia is characterized by having difficulties falling or staying asleep. And this one, a pretty big neurotransmitter that's usually like kind of tied in is GABA. When you don't have enough GABA, you don't have enough inhibitory neurotransmitter helping you to like inhibit your muscle movement and like just kind of slow down your brain to go to sleep. And then some causes of insomnia can be stress, anxiety, depression, noise, just anything that's like discomfort, uncomfortable. And then some effects of this can be mental health conditions, lower performance, tiredness. Because, like we said before, sleep is vital to function. So, if you're not getting proper sleep and rest, you're not really able to function at your peak capacity. And then another disorder is narcolepsy. So narcolepsy is the sudden urge to fall asleep. And I don't know if y'all have seen like Mall Cop. I'm not sure if that's like beyond your, I'm not sure. But in that movie, the main character has narcolepsy. And this is when a specific hormone called orexin, this is what's secreted to help keep you awake. But during narcolepsy, orexin is completely destroyed. So you're unable to stay awake and you just fall immediately into REM sleep at random parts of the day. And it's a hypothalamus related disorder. And it can lead to problems in your daily routine and dangerous situations if you're just falling asleep randomly. Then there's sleep apnea, and this is when you have issues breathing during sleep and you're start and stop. And some causes can be obesity, narrowed airways, and then this can lead to people having daytime fatigue or heart and blood problems. And then night terrors. So this is when people wake up and they're like extremely afraid. And it's related to your REM stage, non-REM stage 3 sleep. And some things that have to do with this or causes can be age, like younger people have more, lack of sleep or stress, and then fever. And then this can lead to fatigue, injuries, disturbed sleep. You're just not comfortable. So now we have an FRQ practice for y'all.
I think, again, for the sake of time, sorry, for the sake of time, because we experienced that interruption, we'll be moving directly into Unit 3, although Unit 2 is a big one, so we encourage you to review these slides later and get that FRQ practice in. It's really important. But for the sake of time, if you are all okay with it, I think we're going to move into sensation and perception.
And yeah, you all should have access to this slide deck, so you should have access to this FRQ. And then we also have a rubric for scoring the FRQ in here, as well as a 5-able team mock response in that study packet. So you should have all the resources to do this FRQ anytime you'd like as well. But we just want to make sure we deliver for you guys all the information that we can and give you all the things that aren't on our slides.
Also, I was going to say we're going to skip our current break. So if you need a break right now, please feel free to take one. Remember that this is recorded. You'll have the recording to return to, and you also have access to the slides. So although we're going to skip the current break right now, just because we missed out on so much time, take it if you need it.
But for those of you who won't be taking a break, welcome. We're going to be talking about sensation and perception. That's always really important, although it does comprise the smallest proportion of the exam. I saw a lot of people in the chat earlier being like, I don't understand Unit 3. That's okay. I'm here to walk you through it right now. So we can get started with the fundamentals of sensation. First, we need to distinguish sensation from perception. Sensation, you can think of it as a very physical process. It's the beginning elements of the sensory system. When you think of any of your senses, any of your 5 senses, the physical stimulation of those receptors, before you interpret any of the information, before you give any of it meaning, that is what sensation is. Your receptors receiving information from the environment. So one example that I have for you is with the sense of smell. Sensation defined here would be the stimulation of your olfactory receptor neurons, where the neurons in your nose. Before you can say, oh, that smells like cereal or that smells like pasta sauce, the active physical stimulation of those receptors, that's what sensation is. Sensation goes hand in hand with transduction. In fact, you can almost sort of think of them as synonymous in their physicality. Transduction is the physical process of the stimuli being converted, the environmental stimuli being converted into an internal representation or neural signals by your nervous system. This is how we communicate between the external world and our internal world before sending those signals to your brain to interpret them. And we can talk about sensation in a few different ways. First, with the absolute threshold, which was discovered by Fechner, he had a few different ways of measuring this. But the absolute threshold is a principle of minimums or maximums. And it's the minimum stimulation needed to detect any stimuli half of the time. This might just be words right now, so let me give you an example. Let's take the sense of audition, of hearing, and along the dimension of pitch. I'm going to play a very, very low pitch, and we're slowly going to increase the pitch of the sound until you can hear it. And then we're going to do that again and again, and again and again. And we're going to see exactly what hertz is needed. What is the exact level of the pitch needed for you to detect that stimulus half of the time? And you hopefully can understand why this principle is called the absolute threshold. We're looking for the absolute minimum needed for you to detect this. Now we can juxtapose Fechner's absolute threshold with the difference threshold, or just noticeable difference. This isn't a principle of minimums or maximums. Instead, as the name implies, it's a principle of difference. And we're trying to understand what is the smallest difference needed for you to detect the difference between two stimuli. So let me give you an example here. Let's say that I blindfold you, have you hold out your two hands, and I place 200 grams of coins in each hand. And I slowly increase the weight in one of your hands, 1 milligram at a time. And I say, tell me when you feel like there's a difference, when one hand is heavier than the other. The minimum needed of the difference in milligrams that I added to one hand for you to feel a difference between your two hands that started out as equal, that would be measuring the difference threshold for the weight of these coins for you. And then again, we would repeat it, and then find that weight in milligrams that you would need to detect the difference about half of the time. Hopefully you can understand the difference. I think the just noticeable difference threshold says what it is, really. When talking about the difference threshold, we can talk about Weber's law. He basically says that to perceive any difference, that difference is going to differ by a constant amount. It's going to be a proportion of the original stimuli. And finally, signal detection theory takes into consideration not only what sensation is and the things that you are going to be sensing, but also other cognitive functions. Like, the idea is that you have to detect a signal among a bunch of background noise, potentially things that could be distracting you, as well as the role of top-down processing in this. Like, what expectations do you bring to this? What attention are you able to allocate to what you're trying to detect versus things that could potentially be distracting you? And signal detection theory brings all of this together, saying that sometimes we can detect the true signal and allocate our attention correctly, and sometimes we can't due to various distractors. We just cannot pick out the signal among the noise. So Pep has a question for you. You can type it in the chat. How do the absolute threshold and difference threshold differ in terms of sensation? Really, we touched on it. Absolute threshold is a threshold of minimums. The just noticeable difference or difference threshold isn't so much that, it's how much do you need to feel a difference in the sensations. All right, and we're going to do some practice with signal detection theory right now. You can type it in the chat. We're going to be labeling this diagram with hit, miss, false alarm, and correct rejection. So this is going to demonstrate, like signal detection theory states, when sensing things, we have to pick up the correct signal among a bunch of noise, a bunch of distracting elements, if we can allocate our attention correctly. So let's say that the signal is present and the observer does respond. What would that be? You can type it in the chat right now. I will reveal the answer in 3, 2, 1. That would be a hit. I don't have the chat open right now, but I trust that you all got it. So
They did.
nice, that's great to hear. We're gonna move on to, let's say that the signal is absent, but the observer responds that the signal is present. What are we going to label that? False alarm, correct rejection, or myth? Correct answer in 3, 2, 1. Nice. Hopefully you guys all got that. That would be a false alarm because our subject indicated they heard something, they sounded the alarm system, but there was actually no signal present. And now we are going to say, I believe that if the signal is present, but the observer did not respond, what is that going to be? We have 2 options left, either a miss or a correct rejection. Correct answer in 3, 2, 1. This was going to be a miss. They correctly rejected that the signal was not present. And finally, our last box is going to be a, sorry, I misspoke. That is going to be a miss. If the signal is present, but they did not indicate that the signal was present, that's going to be a miss. And finally, if the signal is absent and the observer indicates that the signal is absent, or that they don't indicate that the signal is present, that would be a correct rejection. All right, and I think that we are able to move on to our MCQ practice now. What term describes the point at which a person can perceive a difference in brightness in a light, but only just barely so? The key word here is going to be difference. And if you guys can answer that in the chat and Crowdcast, or maybe in a poll, that would be great. And Victoria, just let me know when we can reveal the answer. So you're looking at the Crowdcast.
Yeah, a lot of them are answering in the chat. We're getting a lot of Bs.
Cool, cool. That is actually the correct answer. That's great. It seems like you guys understand the just noticeable difference, and that's amazing. So I think we can move on. So we talked about the first steps to the sensation perception system by going over sensation, and now we're going to talk about the principles of perception. Like I said earlier, perception has to do less with the physical simulation of the receptors, but what your brain does with that information. Once the signals are sent all the way from your peripheral nervous system to your central nervous system, when your brain is interpreting this information, that is what perception is. Think of it more as like the higher order processes. It really is exclusive a process to your central nervous system, whereas sensation incorporates your peripheral nervous system. To throw it back to what we were talking about with Unit 2 earlier, I think that's a great way to distinguish them. So I think I was giving an example with smell. Sensation would be the stimulation of your olfactory receptor neurons, whereas perception would be like, okay, this we smell these chemicals and these chemicals and these chemicals, and we send that information to the brain, and the brain is going to interpret that as spaghetti sauce. That identification, that recognition and categorization of that sensation is perception. And we don't always do bottom-up processing when we go into perceive things or when we go through perception. The role of top-down processing can really be seen with what prior experience and expectations we bring to the world when we set out to perceive things. And this can be summarized in a perceptual set, which is a mental predisposition based on your experience and expectations to perceive one thing, but not another. And I think the example on the slide is a really great example. When you hear someone say something similar to your name in a crowd, and you turn your head and reallocate your attention in that direction, because you bring this mental predisposition to recognize your name, because you've essentially been conditioned all your life to respond to it. So let's say that Ria is in a crowd of people and someone yells out the name Maya, but Ria can't really hear the first letter. So she hears the Aya, and so she turns her head and thinks that somebody is calling her name because Maya and Ria sound sort of similar, especially with the end. This would be demonstrating the perceptual set with your predisposition to perceive your name, especially when called in a crowd, and to respond to that. Perceptual sets are also related to schemas, which are essentially like blueprints or ways that we are able to categorize the world based off of our lived experience. They are really helpful for organizing and interpreting information, because if we stored everything that we perceived as raw data, we would not be able to function at all. So the example on the slide gives birds as the schema or prototype. To be honest, don't worry too much about distinguishing the two. Schema is a little bit more broad than a prototype, but don't worry too much about the difference. So our schema for birds would be that they're like a winged animal, they have feathers, they can fly. My example for you is that I have taken so many AP exams that I have a schema for what an AP exam should be. I know that we're using like the white lifetime tables. I know how to behave in that space. You have to be quiet. I know that I have to bring my number 2 sharpened, like the yellow pencils, because they're not going to let me use the mechanical pencils. So the schemas not only influence our perceptual sets, but also top-down processing, and really are the sum of our lived experience in a comprehensive sort of organization. And they allow us to make expectations based on our lived experience for how the world should be and how we should behave. And they drive cognitive processes. They're really, really important. I already touched on top-down processing, so I'll just elaborate a little bit. Top-down processing is processing that starts in the brain with input from prior knowledge and expectations. This has to do, like I said, with schemas and perceptual set. And another key term that we can tie in here is the context effect. So the context effect is the idea that our perceptions are influenced by our environment, because we bring different expectations of and associations with different environments. So cake sometimes maybe seems to taste better on your birthday than a regular day. And that is because we bring this expectation of what a birthday should be, that it's usually a happy time, and that can lead us to perceive the cake as tasting better. So this would be the role of top-down processing and perception. Perception also relies on bottom-up processing. And like we were speaking earlier with sensation and the very physical stimulation of your receptors, you can think of those sensory processes as being very, very tied to bottom-up processing. This sort of processing, also called data-driven processing, starts at the receptors and works all the way up to the brain. So let's say that I'm looking in a field and I see this red thing off in the distance, and then I get closer and I'm able to see that it's sort of round and it has contours and it's quite shiny. And then eventually I pick it up and I recognize it as an apple. All of that processing, from the color, from the shape, from the contour, from the shine of that object, that would be bottom-up processing in my visual system, happening through my lovely sense of vision. Up until I'm able to categorize that as an apple, that's bottom-up processing. My categorization of that fruit as an apple would actually be top-down, because I'm bringing my prior knowledge of what an apple is to that experience. And so that example shows you that a lot of perception is the work hand in hand of bottom-up and top-down processing. They go hand in hand, but they're both necessary. Yeah, I think that we are able to move on to our next slide. So which phenomenon best represents top-down processing during perception? I'll let you answer that in the chat in Crowdcast, and Victoria, just let me know when we are good to go. Great. Okay, so I will take that many of you have answered in the chat, and
Yeah,
we
we're
can
getting
go
a
ahead.
lot of B's and a couple of D's as well.
All right. All right. So the correct answer is B. It's good to know that we got a lot of responses that were correct. This is top-down processing because you're able to identify a song from just a few notes. And once you have those few notes, the top-down processes of your prior experience, a little bit of expectation maybe, come in. And that's why you're able to identify it so quickly. And so B is the correct answer because it shows the role of experience in this identification process, which is related to top-down processing. D, feeling overwhelmed by loud noises at a concert, that really doesn't have to do with directly bringing prior experience or expectation to this. It's a little bit of a red herring answer, I guess, and College Board does like to do those a lot, so it's good to have practice those. Okay, I think we are now able to move on to our next topic, which is attention, like I was talking a little bit earlier with signal detection theory. There are 3 main points that we want to hit here. First is selective attention, and this is when you are choosing to attend to something with your willpower among many other things that are possibly drawing your attention in. This is a voluntary and intentional process, and you are choosing to focus on one thing. So like listening to a single person talk in a loud room or a single person talk in a cocktail party. My favorite example is me actively choosing to listen to my prof when she lectures and not go on my phone, not look at the game of 2048 someone's playing in front of me, but selectively attending to my prof because I have decided to do that with intention. Inattentional blindness is when we don't notice something new in our field of attention. An example would be not noticing a stop sign while driving by because you were distracted by a conversation. Another very classic AP Psych example, if you look up on YouTube the inattentional blindness video, if you were shown a video of people passing a ball and asked to count how many times they make a pass, and a gorilla were to moonwalk through the screen, most people wouldn't even notice the gorilla because they were so focused on, so they've chosen to selectively attend to the ball passes. If you haven't seen that video, I highly recommend you look it up. It's a good one. It got me the first time, and I was surprised. Unlike inattentional blindness, which is the appearance of something new in your visual field, change blindness is a little bit different. It's similar but different, so this can be a little tricky. Change blindness is failing to notice a significant change in one's visual environment. It's not something new coming in and out, it is a change. So think of that meme where it's in the office and it's like corporate needs you to identify the difference between these 2 pictures. That's what change blindness is. And this, you can actually see it exemplified with movies really well. With the Wizard of Oz, if you go and Google, there are clips, if you hold them side by side, just frames apart. In one frame, Dorothy's hair will be long, and the next one, Dorothy's hair will be short. But most movie watchers don't notice this difference because of change blindness when watching The Wizard of Oz. And because we fail to notice the changes that are just right in front of us in our visual system, even though we are attending to them. Okay, I think that we are able to move on to our next sensation and perception topic, which is the Gestalt Principles. Great. So there are 6 principles that we're going to go over today. Most of them are pretty self-explanatory, so I'll try to move through them quickly, but you can let me know if I need to slow down. The main motto of Gestalt psychology is the whole is more than the sum of its parts, and these 6 principles really convey that. In the chat, juxtapose Gestalt psychology really quickly with structuralism for me, because structuralists would say that, oh, we can break down the mind into its constituent parts, but Gestalt psychologists say, no, it's a little bit more than that. Our perception is influenced by things like expectation, and what these principles convey is exactly that. So first, with our principle of proximity, things that are placed close together are considered to be more related and will be grouped just by how human perception works into their own groups. And you can see that here with the left and right group. Next is the principle of similarity. So units sharing in some similar characteristic, you will automatically group them together as being their own units. And that's why we are perceiving two groups of the teal and the purple and not 12 dots. Next, we have the principle of continuity, that when units are aligned together, you're not going to perceive 7 dots. You're perceiving one elongated line that is continuous. And a little bit more on this, there's also the principle of good continuation. So let's say that we were to have a white box obstructing this line of dots, you would automatically assume that this line would continue behind whatever is obscuring it. That is another related principle. We also have the principle of connectedness. So units that show some sort of connection are perceived as being cohesive units. So here we don't see 12 individual dots with lines in between them. We see them as 6 coherent units. Our second to last principle is closure. And this is where our mind is able to fill in the gaps, and we perceive an enclosed form, like in the example of a circle here, and instead of 5 individual lines. So I think this really gets the point across more than any of the other principles in this example, that the whole is more than the sum of its parts. And you have to ask yourself, why am I seeing a circle instead of 5 different lines? And that's because of these expectations and also this tendency for the human mind and perceptual systems to simplify what we are seeing into coherent forms. And our final principle that we talked about a little bit earlier is figure ground. Again, with the simplification, it is a tendency for the human mind to simplify what we are seeing into a figure and a ground based on we bring a little bit of prior experience because we need to be able to assign meaning to what is in the going to be the figure as opposed to the background. And actually what we have here is a bistable figure. You can see either 2 faces or a vase in the middle. And if you look at it long enough, it should switch. And that's because there are 2 different ways to perceive this. But the main point here in terms of the Gestalt principle is this figure ground, that when we look at things, we automatically want to simplify the scene that we're viewing. We want to find a meaningful figure that sticks out against the background. All right, and I think that with that, we are able to move on. I think Pep might have a question for us now. So chat, how is perception influenced by contexts such as culture? Victoria, you're welcome to hop in on this one if you want. I'll let you guys get typing in the chat. But while you do so, I'm going to keep on talking so you can listen to me. Obviously, for most of us, this visual illusion gets us. We think that the top line is shorter than the bottom line. But if you were to take out a ruler and measure it, you would actually be able to see that the lines in between those corners are actually the same length. And why is that? And specifically, what does culture have to do with this? So with this illusion, they did go ahead, Victoria.
No, I was gonna just say this kind of goes back to what we were saying before about the cultural perspective, where our different perspectives on life and our different way that we were raised and how we interact with society can definitely lead to different interpretations in our thought processing.
Yeah, exactly. And that's why like a sociocultural perspective in psychology is so important. And speaking of that, researchers did a cross-cultural study comparing those who grew up with around buildings that had architecture with corners and right angles, and compared that to the results of people who are looking at this illusion who didn't grow up in environments that had corners and right angles. And what they found is those who didn't have an environment with such corners growing up actually were not fooled at all by this illusion. And so that really speaks to they were able to see the lines as the same length, and that speaks to the role of culture in our perceptual systems. And with that, I think we can move on to visual anatomy. Great. So the eye is a huge deal. It's probably the most important human sense. The amount of cortical space in your brain that is afforded to just your retinas alone is actually so crazy. So we're going to break this down for you because it's such an important part of the human body and the perceptual experience. So there are about 7, just kidding. Yeah, there are about 7 parts of the eye that are really important for you to remember. And you can remember the order in which light travels through the eye, the order that these parts of the eye sort of are taking place. With this handy dandy mnemonic, can people in London read French? Oui. So now I'm going to break down what each of these parts really stands for. So with the C, that C stands for cornea. Cornea is the outermost layer of your eye. It's a layer of thin tissue. It really, to be honest, doesn't do much except for protect your eye. And I remember this with this weird little saying that if my younger brother threw a piece of corn at my eye, my cornea would protect me if I didn't like blink or anything, because that's really all the cornea does. It protects your eye. Next, we have the pupil, which is a small opening in the eye, and it's controlled by the iris. The iris is the colored part of your eye, and it can either constrict or dilate depending on how much light is in your environment to let the appropriate amount of light in. Again, the iris is the colored part. That's really important. You can see that in our diagram. Our iris is pointing to the blue thing in the eye. So this person probably has blue eyes. After light passes through the pupil, which is either going to be constricted or dilated to let in the right amount of light, the light is going to pass on through the lens. The lens is important because, like a camera, it makes sure that the light in the picture is in focus so that we can have a clear picture which is going to be projected on the back of the eye or the retina. The retina is where all of our photoreceptor cells are located. This is where transduction is going to occur. This is why we can really see everything that we see. The retina can be broken down into 2 parts, only one of which is on our diagram right now. The first part is the center of the retina. That's called the fovea. A lot of, or actually, cones are located in the fovea. No rods are located in the fovea. Don't worry if you don't know the difference between a rod and a cone yet. We're going to get there. Just be patient. Trust me. But the fovea here has great acuity. It's really great for picking up details. Like if you're reading a book, that's all the work of your fovea, as well as being responsible for color vision. So if you're reading a book and then you look up and you see like a blue wall, the reason that you can both read and see that your wall is blue is because of your fovea and the cone cells located there. Just quickly, the other part of the retina is called the periphery. That's where a higher concentration of rod cells are located. There are some cone cells, but not as many. So the periphery is important for sensitivity in darkness, as well as black and white vision. And finally, once that we have the photoreceptors have transduced the light and the energy from the light into an internal neural signal, that signal is going to be sent out of the eyeball to the brain via the optic nerve. And looks like that we already have our diagram all nice and labeled for us. So I think we can move on to an MCQ practice now. Oh, just kidding. Pep has a question for us, but we've kind of already gone over it. But type in the chat right now, beginning at the retina, describe the pathway visual signals travel from the eye to being processed in the brain. And quickly, so from the retina, it's going to go to the optic nerve and out of the brain through the blind spot, and then it's going to go to the primary visual cortex in the occipital lobe. That's where it's going to go. Also, it's going to pass through some subcortical structures too, but I left those out. Sorry. Okay, MCQ practice time. Which part of the eye's anatomy is primarily responsible for fine-tuning the focus of light onto the retina? You can let me know in the chat. It seems that we have the chat going right now. Great. Okay, I can see that a lot of us are answering D, and that is the correct answer. Nice. Very good, everyone. I saw some Bs as well. I understand that eye anatomy can be a little bit confusing. The lens is really what's going to focus the light on the retina, so we have a clear picture. Iris controls how much light gets in to the eyeball, and the iris is going to occur in our pathway of the eye before the lens does its work. Okay, so now we can talk about the difference between rods and cones. Oh, I'm seeing in the chat, can someone write the acronym for the order of the eye again? The acronym is Can People in London Speak French? Oui. And that'll get you covered for the order of the eye. So there are two different types of photoreceptor cells. Like I said earlier, we have our rods and we have our cones. Our rods are what detect black, white, and gray tones. And you can remember this by this little mnemonic device, X-ray and rods. Both of them have R's. Think of them like that. There are about 120 million in the retina, and they're very light sensitive. Generally, they're more sensitive than cones, but they're specifically very helpful for seeing in the dark. And they're also needed for peripheral vision as well. And there are no rods at all in the fovea, although there are some cones in the periphery of the retina. We can contrast rods with our other type of photoreceptor cell, which is our cones. Our cones are responsible for color, like the cyan color, the cyan cones. It's a great way to remember it. Another way that I like to remember it is just by going off of the semantic meaning of cone when it isn't a photoreceptor cell, like a giant bright orange traffic cone. I can remember that the cone is bright orange and cones detect color, and the bright orange reminds me that cones do detect color. There are about 6 million cones in the center of the retina, or the fovea, which is really the home for most of the cones. And cones work best in daylight. They have great ability to detect detail, although they're slightly less sensitive than rods. And they're absolutely necessary for being able to do a lot of your daily functions that include color vision, but also apart from color vision, like being able to drive, being able to read a book, to do your homework, to study for your AP exam. You can thank your cones for that. And next, we're going to talk about a place where we actually don't have any cones or rods, which is going to be our blind spot on the next slide. Or not. I will start talking about the blind spot now. That's okay. So in the blind spot, we don't have any rods or cones or photoreceptor cells because this is where the optic nerve is leaving our eye. If you actually look at the construction of the eye, it's really amazing that the eye works because it's constructed in a little bit of a backward state, but we don't have any photoreceptor cells where the optic nerve leaves the eyeball to go to the brain to tell us about all of the beautiful things that we're seeing in the world. Because of this, we have a blind spot, but we don't really perceive there being a blind spot in our visual field due to the higher order processes of perceptual filling in. And another important area of the brain that I would another important area of the retina and of the eye that I would like to hit again is the fovea. Again, would be on the next slide if we could get to that. If not, no worries. The fovea is responsible for color vision because cones are concentrated in this area.
I'm gonna jump in real fast. Hey, Sean, if you are listening, can you change the slide, please? If not, then I will try to take control, because maybe we lost Sean or something.
Cool.
Okay,
Thank you so much.
To take control of screen sharing. Just give me a minute and keep going.
Okay, cool. There's not a ton on the fovea that I haven't said already, but the cones are going to be concentrated in the fovea. This is what's going to be responsible for your ability to detect color, as well as your ability to detect fine details in everyday objects. Yeah, so let's say that I am actually in the chat. Can you tell me what other everyday activities are needed for you to, what other everyday activities necessitate your phobia? Like, there's reading, driving, studying for an AP exam. What else? You guys can be creative. Drawing, that one's a good one. Art, I like that one. Those are
Art?
good.
Video
games. Typing, that is so true. Typing. Schoolwork, yes. Athletics, that is for sure. Okay. So, again, what type of cells make up the structure? I know you guys got it. Just quickly type in the chat, and then I think we are ready to move on to our depth perception with our monocular and binocular cues. Okay, so there are two different ways that we are able to perceive depth. One occurs with only one of your two eyes. The other one occurs with both of your eyes, and it necessitates both of your eyes. With only one eye, these are called the monocular cues. And there are three that I want to touch on first. One, relative height. So if we look at this picture, the one with the duck, the one on the top middle of the slide, you perceive the house as being further away in this picture from you than the duck, because the house is located higher up. It has more relative height compared to the duck, which is located more toward the bottom of the picture. And this is relative height. Objects higher in the scene are perceived as being farther away from you. Another monocular cue is linear perspective. And this plays off the idea that we perceive points, if we look at two parallel lines, eventually we perceive them as being really far away from us when the lines begin to converge into one point. We're called the vanishing point when we look at it far enough. Now look at the foreground of the picture, we're able to see two independent lines that are quite far apart. But if we look at the background of the picture, we see them converge into one. Now this is the work of linear perspective. And again, this tells us about depth, not only about depth, but also relative size as well. And there are different visual illusions that you can look up to play on this. And again, you only need one eye to be able to use this depth perception cue. The final monocular cue that I want to talk about is interposition, which is laid out pretty well with the picture on the bottommost picture on this slide. This says that if an object is obscured by another object, you can see the circle is in front of the square. The square is going to be perceived as being farther away and a little bit smaller, I guess, than the circle, because the circle is in front of it, it's obscuring it, and we perceive the circle as being closer to us. So to summarize, if an object is covering another object, we're going to perceive it as larger and closer to us. If an object is being covered, it's going to be perceived as farther away and a little bit smaller. Again, these are our monocular cues, and we do only need one eye to be able to perceive depth in these ways. And so that should tell you that people who are only able to see out of one eye, do to some extent have some ability to perceive depth, albeit significantly less than people who are binocular. But there do exist some cues that work for them. Binocular cues, on the other hand, necessitate both eyes to be able to detect depth. And here we have retinal disparity, which is essentially the difference between the two images that are on your retinas. Your brain will compare the disparity between them and determine how far away an object is, depending on the images that are on both of your eyes. So if you just hold out your thumb and then switch your eyes back and forth, close one of your eyes and open the other eye, and hold your thumb out very far away from you and then bring it close, you should be able to see that you have an increased disparity when there is an object close to you. And these differences, especially differences up close, of the two images on the retina, tell your brain and provide cues about depth and how far away something is from you. Our final binocular cue is convergence, which is the idea that when let's hold out our thumb again and follow your thumb as you bring it close to you, your eyes are going to converge. They're going to turn inward when you need to focus your thumb when it comes closer to you. And this is the idea of convergence, that your eyeballs are going to need to shift inward to focus more, and your brain, evaluating the extent to which your eye muscles need to change to focus inward, that is going to provide an additional cue of how close or far away something is, cues of depth perception. All right. And Pep has another question for us to explain how depth cues contribute to our perception of three-dimensional space. Victoria, if you want to add anything in on this one. I love Pep and all of his little questions. But yeah, essentially a TLDR version of it. Depth cues allow us to assess how close or how far something away from us is through a combination of molecular cues that only use one eye and binocular cues that use both eyes. Yeah, if you want to add anything, go ahead, but if not, we'll just keep going right along with our color vision theories.
I just wanted to pop in and say real quick for those that haven't seen it in the chat, we are going to stay about 15 minutes extra to try to finish this. We are pretty close to being done with content, and then we will switch over to doing our cram quiz.
Yay, okay, we're in the final push, guys. So there are two theories of color vision. The first one that is called trichromatic theory, and this states that color vision is determined by the relative activation of three receptor mechanisms that are specialized to detect red, blue, and green. And any color in between that isn't red, blue, or green is perceived as the relative combination of the activation of these three receptor mechanisms. And actually, this aligns with the idea that we have three different types of cones that are each specialized to detect maximally the wavelength of red light, the wavelength of green light, and the wavelength of blue light. And for a really long time, trichromatic theory of color vision was super, super popular. But one thing that it wasn't able to account for was the behavioral evidence of the after image effect. You guys may have experienced this before when you stare at a colored object for a really, really long time. And then you experience essentially sensory adaptation, and then you look away onto like a white wall or something, you'll see the negative after image of the image that you were seeing before. And trichromatic theory couldn't explain this, but opponent process theory can. This is the second theory of color vision, and it states that color vision is determined by the activity of two opponent mechanisms. There are three pairs of these opponent mechanisms that you are going to need to memorize for the exam. And opponent process theory does explain the after image effect. It's a little complicated. I could explain it more at the end if you wanted. But let's say that you are staring at something, a stimulus that is like blue for a really long time, and then you look away. The neurons that are specifically responding to that blue are going to become fatigued. They're going to fire less. And once you look away, you're going to perceive yellow. And this is because these colors are opponents and complementary. And so they're linked in pairs of three like this. One way that I liked to remember this for the AP exam was that the pair of green and red, they're like Christmas colors. So that's one pair, the Christmas pair. One is black and white. That's the monochromatic pair, easy. And then blue and yellow. These colors might be meaningless to you, but my high school's colors were blue and yellow. So you can think of me going to Foothill High School and my school colors if you need to memorize this for your AP exam. So the school colors of Jessica's high school were blue and yellow, and that is the final pair. It's a little bit complicated, and actually both of these theories are correct. Like I said earlier, we do have three different cones that respond to maximally respond to three different wavelengths of light. So that part is true, but also in a way, I will spare you all of the biology for this. The opponent process theory is also true because in these neurons, one part of the cell will be inhibited by blue and the other part will be activated by sorry, the one part will be activated by yellow and the other part will be inhibited by blue. So it's a little bit complicated. Again, I can explain more in the Q&A if needed, but these are simplified, the two main theories of color vision that you will need to know, and they are both correct. Component process theory more on the receptor level, and trichromatic theory more in the higher areas of cortical processing. All right, we are almost done. We're in the home stretch. I think now we can move on to our anatomy of hearing. And we can compare the trichromatic theory and opponent process theory in the chat. I'm going to go through hearing quickly. Hearing, there's a little bit less anatomy and it's less complicated, as well as the rest of our senses. So hearing first, we're going to start with the outer ear, which is composed of the pinna, which is like literally the outside of your ear right here. Like the thing that Van Gogh cut off, to be honest, it doesn't do much for hearing. I guess he was okay. So the pinna is this outer area of the ear. It's really important for localizing sound, but beyond that, actually detecting sound existing doesn't do a whole lot for the sense of audition. So the sound is going to travel through the pinna, through the ear canal, to the tympanic membrane or the eardrum. This eardrum separates the outer ear from the inner ear, and it serves a function of somewhat amplifying the sound wave because the sound wave is going to hit it and cause it to vibrate, and it'll amplify a little bit the sound coming between the outer ear and the middle ear. The middle ear is composed of the three smallest bones in your body, the hammer, anvil, and stirrup. These are also called the ossicles. And these ossicles serve a very important function of really amplifying the sound waves, because once you transition into the inner ear, your inner ear in the cochlea, there's not air in there. The sound waves aren't going to be traveling through gas. It's actually going to be traveling through liquid, because your cochlea contains liquid. And if you've ever tried to listen to sound underwater, it's a lot more muddled. It's a lot more difficult to hear compared to when you're out in the air. So the ossicles need to amplify this so you can still continue to experience that sound wave with at least some intensity to be able to sense and perceive it. So in the cochlea, this is where transduction occurs via your hair cells. These are the actual receptor cells that are being stimulated to change the mechanical deformation, the energy of the sound wave traveling through the air into a neural impulse. All right, and now that we've traveled from the outer ear to the middle ear to the inner ear, I think that we are able to move on to our final two senses. Oh, what structures are responsible for vestibular sensation? That is important. If you want to type that down in the chat, what vestibular sensation is first, and to what structures are responsible for this. I think that I will have to move on for the sake of time just to be able to finish this in our final two senses. But vestibular sense is important. You would probably like fall over without it, to be honest. Finally, our last two senses that we're going to cover tonight are taste and smell. Taste, what you need to know is that your taste buds contain clusters of taste receptor cells, and they are tuned to be able to taste 5 main different stimuli. They're able to distinguish between sweet, sour, salty, bitter, and savory, or umami flavors. Umami was the last one to be added. And all of this information goes to the gustatory cortex in your brain. Our final sense for tonight is smell or olfaction. Smell is a really interesting sense. It's not like the other senses in a lot of ways. But the receptors that you need to know for olfaction are the olfactory receptor cells. These are contained in the olfactory epithelium in the nasal cavity. And these detect the odors. These are stimulated by the chemicals in the air. This is where transduction occurs. Then this signal is sent to the olfactory bulb, which gathers information about the different chemicals in the air and the different odors, and then sends the information to the brain in a sort of mapped out state, and it's going to go to the piriform cortex in the brain to be perceived and processed. And it looks like we have made it through, mostly. We're still here, in our seats with our 3 units.
We're going to skip this free response question again. We are going to move straight over to our moving quiz. So we're going to start the cram quiz. I have the QR code for you right here. And I will also throw a link. Actually, if some of my hosts can get on and start putting links into the chat and just continue. Usually we would throw up the bubble where you can click it in Crowdcast, but Crowdcast decided to riot tonight. So fine. Go ahead and use this QR code. Throw the links into the chat team. Perfect. And let's just keep throwing that link into the chat repeatedly as well, so that as people go through, well, 5 times in a row, as people connect a little bit later, yeah, we can do that. So let's get, wow, my 800 tabs. I don't know what screen I'm even sharing anymore. But here's what we'll do. Ooh, what can you guys see? What am I sharing?
We can see the first question of the cram quiz.
Perfect. Can you also see my like
Yeah.
bookmarks? Oh, great. Okay. Cool. Don't copy the link here and go through everything, guys, please, because that's rude. Don't do it. You can see my entire
Cheating as
browser.
well.
Lovely. Luckily, I'm not hiding anything from you. It's just all my emails, all my everything. So go ahead and get started. Love that we can see my whole browser.
Yeah. Also, fun fact for you guys. I see you guys were talking about umami in the chat, our 5th sense. Did you know that umami, or MSG, monosodium glutamate, our ability to sense that is actually based on glutamate receptors. So that's pretty cool. I'm seeing a comment, like an acronym to remember the parts of the ear. To be honest, I was trying to come up with one earlier today and I couldn't. But the parts that you need to remember simplify to PTOCH, P-T-O-C-H. I was trying to come up with something like potatoes teach overnight calculus, and it was hard, except I had to add in some extra words. It's quite a bad acronym, but if you want to use it, I can type it in the chat for you guys quickly. Yeah,
To do that, we are going to reveal our first answer to our first question. We'll do that right now. Can you guys also, not currently, but then, could you hear the cram quiz music? Just wanted to, oh,
I cannot, but I was also talking.
I don't think we were able to hear it, Jenna.
So was I literally on screen dancing like this and nobody could hear the cram quiz music and no one wanted to tell me? Well, that is wildly embarrassing, but that's okay. We'll go to the next question because it looks like there was an overwhelming majority on this one, and I will see if I can turn the music up for you guys.
All right, what is the measure?
No?
You're not able to hear it. That's okay. You can listen to my voice instead as I read you this question. What is the measure of central tendency that identifies the most frequently occurring score in a distribution? We're at a good amount of participants who have responded. You can go ahead and reveal the answer. Wow, with overwhelming consensus, we have the correct answer at mode. That is going to be the most frequently occurring score here.
I'm going to try to stop sharing and then reshare so that we can try to get the audio on, because the audio on these is really, really fun. Okay, I see what I need to do. This one. We will do the audio. Can you guys see my screen?
Yep, we can see it.
Same one.
Oh, there's the audio.
Now we know why I was going like this. So worth the wait, right? So worth it. Michael does have the best studying music. And I do also wish for you guys that the audio was on the AP exam too. That would make it, I think, a lot more fun.
I feel like that's a good example of context-dependent memory. Like you encode with samba music, you perform better, you retrieve better with samba music.
I would agree with that. I think that that is awesome. Okay, so this one there's a bit of a skew here. All right, review this question.
All right, so this question asks, which psychological perspective would most likely examine the role of brain activity in decision making? The majority of you did get it right with the biological perspective. This question, I would say the key words are the role of the brain, and that should scream to you biology. The second most chosen response would be the psychoanalytic theory. This would be more like Freud, his funky pre-association stuff, his your mom stuff, not so much the brain. It would be more personal experience. The unconscious would be a huge vocab word that you should look out for if that were to be the right answer.
Yeah. So just remember, guys, with biological, it really talks about the links between the brain and our nervous system and our behavior. And biologicalists basically try to understand how our neurotransmitters and our hormones kind of influence our emotions and our thoughts and our actions.
The music stopped, or is it just me?
Maybe it'll restart.
What a bummer.
Yeah. You guys can answer a question in the meantime, though. What is the ethical guideline mandating that information about participate will be kept secret?
Did anybody else's sound get distorted or just one person's? Maybe that, okay, nobody else's sound is distorted. There's my notes again. Hi. Okay, so the answer was anonymity, and overwhelmingly, it looks like we got that one. So everybody, fingers crossed, when we hit next question, our samba music will come back. Ready, set. Yeah, that's what it needed. A little hope, dreams. It's because you crossed your fingers and toes. You are so right. Did just hear from Sean. Sean said that his computer completely froze. So what that means is that although the crowdcast gods needed him sacrificed, he has returned to us. I don't know if he's here yet, but his well-being is okay at least. All right, we're gonna reveal the answer. it's the like cessation of the samba music. It's just so sudden. All right. Which endocrine system is responsible for our daily biological rhythms? The pineal gland. It looks like we had a little bit of a distribution with the thyroid and the adrenal gland as well. So with this one, does anybody want to review this one, or do you want me to?
I think you could go for it, Jenna. You're a hormones person.
Yeah, that actually is my work, but I usually work with reproductive hormones and not these ones, but I can get there. So the pineal gland is a gland in the very center of the brain that's responsible for things like melatonin, and other types of biological rhythms being like circadian rhythms, diurnal rhythms. That's what we're looking at when we look at the pineal gland. Whereas when we are looking at the adrenal gland, the adrenal is responsible for stress responses. So things like fight or flight, the sympathetic adrenal medullary system, which is the cortisol response. And then the thyroid gland, the thyroid controls a bunch of other stuff that I actually like literally never done any research on the thyroid gland. So somebody else could probably pop in with what the thyroid does, but not me on that one. But yeah, so the pineal is things like biological rhythms, circadian rhythms. That is our sleep-wake cycle. So that's what we have there. Oh yeah, thyroid is like growth hormones. Thank you all. That is very lovely for you to help me with that. All right, next question. Oh, go ahead, Victoria.
No, I was gonna mention something about metabolic processing. It's very heavily kind of involved with that too.
Perfect. Thank you so much. Okay, cross fingers and toes for samba music. Ready, set, go. We are going to reveal because we are already late and we don't want to stay here forever. Okay, perfect. Overwhelming majority regulates blood glucose levels. Amazing, intelligent, smart, fantastic. Fingers and toes crossed. Ready, set, go. Nice. Somebody in the chat asked what releases hormones. Glands release hormones, and we have a ton of different glands distributed throughout our body. The pineal gland releases hormones. The pituitary gland releases hormones. The reproductive glands, the ovaries, the testes, release hormones. There are tons of glands that secrete hormones. Akin to hormones are neurotransmitters. So you can see our neurotransmitters here in this question. All right, revealing answer. 3, 2, 1. Great job. Excellent, awesome, amazing, fantastic. See, there was a little uptick in GABA. GABA is the primary auditory neurotransmitter. It regulates like sleep-wake cycles. So GABA is going to slow down the body. It's what anti-anxiety meds work off of. So anti-anxiety meds are going to bind to GABA receptors. Moving on. Going to reveal. Nicely done. Great job, you guys. This was, yeah, overwhelmingly. Not even going to talk about it because you guys killed it. Good job. Next one. On this one is electrical activity. Like Ria said, all caps in the chat, electrical. Okay, revealing. Nice. So that's our EEG. Remember that the EEG is measuring the firing and the action potentials of our brains. That's what we're looking at with the EEG. It's got great temporal resolution, so time related. Alrighty. So somebody in the chat asked about the difficulty of these questions compared to why did it stop? Rude. Okay, the difficulty of these questions compared to the AP questions. These questions are much easier than AP. These are like trivia type questions. AP questions more difficult. So keep that in mind, but these are just kind of like trivia based to keep us going. Yeah, I don't know why the music cut off. It clearly heard me talking and it was like, must stop. Thank you. So, you know, what can I say? All right, we're gonna reveal. Oh no. Okay. Somebody is really, really good with sleep and dreams. I think it was Bria, or was it Jessica? Can somebody jump in for the sleep and dreams?
I'll let
Yeah,
Rhea tackle it. I can add in.
Cool.
So for sleep, the one that's most associated with emotional and vivid dreams is REM sleep, because this is when your brain is active, you're still sending out signals, you have those beta waves. It's the most like active part of your sleep cycle, whereas all the other stages, the rest of your brain is mostly like kind of turned off. It's these slower waves that aren't really as active as when you're in REM sleep. So that's why you're gonna have more like vivid and like thought-provoking kind of dreams when you're in that deep REM sleep. Did you want to add anything, Jessica?
So much.
No, you handled it pretty well. Just REM is where you dream.
Perfect. Perfect. All right, moving on. Cross those fingers and toes again, because we didn't do it last time and we didn't get our music. So cross them. Works. It works. The Zoom gods like us. Alexa F, you can take a break to go handle that if you need to. No worries. Revealing the answer. Easy.
Nice.
You guys had it. Excellent job. We're not gonna review this one because you got it. Somebody found the emojis. Only got the one though. You guys can hit the little emojis if you see them. Okay, revealing. Uh-oh. Oh no.
Man. It's all right. We may have gotten you confused with the MCQ question earlier in the slideshow, to which the answer was lens. But this is asking something slightly different. Which part of the eye controls the amount of light, not the focusing of the light, the amount of light entering. That'll be the iris, which determines how wide the pupil is, whereas the lens focuses the light. So think of the lens being associated with focusing, the iris controls by constricting and dilating the amount of light. But still, most of you got it right. So impressive. Good job.
The iris is actually a muscle. So it's a muscle, it's a sphincter muscle, and that is what the color of your eye is. So the color of your eye, this is the iris, and it is a muscle. Oh, my dogs are busting in. Oh no. Moving on. Come say hi to the people. about that.
Jenna, your dogs are so cute. What
Thank
kind of
you.
dogs are they?
I love them so much. Yeah, a lot. God. Okay, we are going to reveal the answer. Nicely done. Great job, you guys. All right. I think we can just keep because that was pretty good. So moving on. I have the questions. Layla said attack of the doggos, which is funny because one of them is named Layla. So I have Layla and Ray. Ray asked what breeds. They're mutts. They're just little shelter mutt dogs, but they are litter mates and they're very cute and I love them. Fatima wants the 5-able shirt. We do love that one. This one came from last year. We didn't get 5-able shirts this year. I don't know why, but I think Jessica also has a 5-able shirt that we got last year. Almost there. I'm gonna wait for just a few more and then we're gonna reveal it. All right. Uh-oh. All right. Who wants to hop in for this one?
I can do it. This question is a little bit tricky. But the key here is that musicians are going to come in with prior experience. They're going to be essentially experts in being able to detect pitch changes at very quiet volume levels. And so you kind of have to assume that musicians will have prior experience and expertise compared to non-musicians. And this is going to be the role of top-down processing as opposed to bottom-up processing, which is not the right answer. Gate control theory has to do with pain. That's really not the right answer. And opponent process theory has to do with vision, not audition. So by deduction, like B has to be the correct answer here.
All right. Last question. You guys ready? Last question of the night. 30 minutes overtime. Thanks for sticking with us. Final question of the night. I hope we're gonna do pretty well. Why did the music stop again? What a bummer. I hope we're gonna do well on this one. Fingers crossed. We'll pretend da 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 da. Whoa, whoa. I sing for you, but I really want to. Just whoa, whoa, whoa. I don't know what it says there. Da 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 da. Okay, a few more, and then we will reveal. And we will be done for our extra night. Okay, revealing answer. You guys, we can do this though. I believe in you. Is it because olfactory is just a word that we are not super familiar with? Maybe that is why. I had faith here. That's okay. We just need to practice. I think this is around where we got a little messed up a little bit with some of the tech. So olfactory bulb is sense of smell. And the next most common answer was temporal lobe. So the temporal lobe is the one that sits right here, right above your ears. And it is much more responsible for like language comprehension, like things like understanding of rhythm and music. That's going to be the temporal lobe. The olfactory bulb is indeed a part of the brain. And then it's going to relay straight to the cerebral cortex. The olfactory sense is the only sense that doesn't go to the thalamus. Every other sense goes to the thalamus first before being distributed to other parts of the brain. The olfactory bypasses the thalamus, doesn't go there. And that is why, if you've ever had that effect of you have a memory or you smell something and it cues you into a memory, but we don't have that really with other senses. It's because the olfactory bulb does not relay straight to the thalamus, so there's no inhibition of that relationship. So there is that. With that, just a couple of reminders. We will be on Zoom again. Sean is in the chat. We will be on Zoom again tomorrow, but please remember to go to Crowdcast to get your Zoom link. Go Crowdcast Zoom link, then we will be Zooming tomorrow. I can't think of any other announcements. Oh, there is our survey. Okay, so please be gentle on us with this survey, remembering that these tech issues were not our fault. I need a moment to find it. If Sean also can find it, because I gotta dig through some chats here to find this one out.
Sean's
We are
got it
going
in
to-
the chat.
Oh, somebody's already got it in the chat?
Yep, Sean got it.
Perfect. Thank you. Wonderful. Please be gentle on us with this survey and know that we did our best with trying to get all of the issues figured out for you guys. We appreciate your time. Get that survey. We will see you all tomorrow.